Instead of just thinking of this as the assembly process, let's break it down into some steps. And you could break these steps down even further, but I think this is enough for understanding the concepts. So, first of all your structural proteins, which are things like the M protein, shown up here, are going to need to be expressed, which just means making the protein from the subgenomic RNA. And that's going to happen in the infected cell. Then there's all of this processing stuff that's going to happen. A lot of it happens while it's being translated or just after it's translated. So things like glycosylation, acylation with fatty acids, phosphorylation, and like folding, refolding, any other post-translational modifications. So that is like making the protein and making it ready to be able to do its job. Next step is going to be transport, and so the proteins have to get where they're going. And this only applies to proteins that are in the membrane. As far as I am aware, proteins that are just in the cytoplasm are not going to be actively transported. They're just going to have to find their own way to the right spots, but probably, there's some other way that they're getting around. So transport is going to involve, in the case of the membrane proteins like M and E and S, it's going to involve moving the entire membrane that they're in. Yeah. It's like in the old days when you had a king, and the king does not walk, but you pick up the king's chair so the king can ride in comfort and, carry them through the streets. Like that. And so there are going to be particular interactions and motifs that are important for that. Not all of which are known, but enough of which are known. Then you're going to have packaging. Packaging is the selective part. So think of the Amazon worker, going and carefully selecting, handcrafting whatever thing you just ordered and putting in the box, putting the wrapping paper around it, like that, like Santa Claus does. Packaging involves specific binding to the genome. And mostly that's going to be done by N, but there is a component of M that also is important for packaging genomes and not just picking up RNA in general. Once you have at least one of these interactions where you have specifically picked up genomes, that seems to be there. That may be enough that you trigger the rest of the packaging interaction to happen elsewhere. But the packaging interaction is going to be dependent on a packaging signal, and that's a thing that's only going to occur in a part of the genome, like a small part of the genome. So the packaging is very discriminatory is what I was going to say, but it discriminates between like genomes of the virus and subgenomics and all the tRNAs and messenger RNAs that are floating around in a cell, which could otherwise easily get tangled up. Then we've got assembly. So this is all the protein parts coming together. And if they're holding on to RNA, great. The RNA will be incorporated, but you can make coronavirus particles without any RNA in them, or without any viral RNA in them. And you can make them without a lot of stuff. You can make them without the N protein. You can definitely make them without spikes. So it's just M, and then E will assist M in its job. So assembly, we're binding all these things together, and so now we've got a flat version of all the proteins with various domains dangling down at the bottom, and it's just going to be sitting there in a membrane somewhere between the ER and the Golgi. All of this assembly stuff is going to happen as it's going back and forth between the Golgi and the ER, and somewhere in the middle the magic will happen, and we will bud into the vesicle that the M and S and E proteins were already on for transport. So it's like it's going to steal the car, or steal part of the car, and make that into a virion, and then ride around in the rest of the car. It's weird. The metaphor breaks down. Doesn't work at all. Yeah, but here we go. So this part of going from flat to bulge, like that is budding. And budding is not separation of the virion from the vesicle that it's budding into. Budding is just forming the little bud, like on a little flower. And then scission is when we cut the bud off, and that is going to be something that's done by the E protein, we believe, using a amphipathic helix, like all the good scission proteins seem to do. Okay. In older literature, you'll read about a protein called hemagglutinin esterase. It's on a few coronaviruses, like the mouse coronavirus. It's not on SARS coronavirus 2, and it's actually not that common in coronaviruses. When it's there, it's a secondary attachment protein, and it will bind to sialic acid, which is something you would find somewhere on the surface of most glycoproteins that would be on the surface of the cell. So it makes the virus stickier, it makes it stick to cells better. The hemagglutinin part sticks it to cells. The esterase part actually cuts off the sialic acid after a while. So it's like a temporary attachment method. And then if it doesn't find what it's looking for in terms of an actual receiver, then it can move on to the next cell. All right, let's look at the parts of this process. First of all, these are the proteins that are involved. So we've got our M, N, E, and S, and they're always in the order S, E, M, N. So I don't know why I drew them that way. I drew them that way because they're that way in the little drawing. That's why. Yeah, I'm thinking the order they are in the genome. Anyway, so yeah, M's job is going to be like the glue. It's going to bind, it's going to sit in the membrane. It crosses the membrane 3 times, and it's going to interact with both N and S, and it might also interact with E. Probably does, but it's probably not like they stick together and stay together for a long time interaction. It's just they're like attracted to each other and then they break up. Yeah, you know people like that. Yeah, over and over and over again. Nucleoprotein is going to interact with the viral RNA and other nucleoproteins and M. And nucleoprotein is going to be a dimer, as is the M protein. They're going to work as a dimer, so it takes 2 of them to make the machine that does the job. Envelope protein is going to interact probably with M. Yeah, there's a probably in there that I didn't write. And it is going to be an important factor for scission, although you can knock it out from several coronaviruses with very little effect to no effect on the virus efficiency. So it's like a minor efficiency aid. It helps out with scission, but there must be other ways to do scission that don't involve E. Probably involves some host protein, and I just can't give you any more details on those at the moment because I don't know any. Then we have our spike, and spike is going to work as a trimer. That's this great big thing that's going to stick out from the virus, and spike's going to have a transmembrane region that's going to interact with the M protein. And so spike doesn't directly interact with N or RNA, but spike interacts. The spike bone is connected to the M bone is connected to the N bone connected to the DNA bone. There you go. That's how the song goes. If you don't know that song, that's okay. It doesn't matter. That's just how they're connected. All right, let's do a little more on the properties of these things. So let's talk membrane proteins and non-membrane proteins. How are they modified by the cell, and why are the modifications important? So the M protein has a little bit at the N-terminus, like the front of the protein, that actually is going to stick out of the virion. The problem I have with this is that there should be about 1,200 of these M's on the virus, and so on half of virus you should expect to see about 600 of them, and I can see about 30. Yeah, so they're a little sparse on the M's. Yeah, the number of spikes is actually okay, but oh well, can't win them all. The M's are going to be glycosylated usually, although we don't know this for certain yet for SARS-CoV-2. Has not yet been shown for SARS-CoV-2 or SARS-CoV original. But yeah, in the viruses where it's known that are beta coronaviruses, it is an O-linked glycosylation, so to a serine or threonine and to the hydroxyl group at the end of that. So different from N-linked glycosylation, slightly different sugars in some cases, hooked up in a slightly different way, but they kind of do the same thing. And it's going to be a shield from the immune system. It is going to prevent antibodies that otherwise could have bound to that sticking out part of M, and potentially cause the virus to be not infectious, or just phagocytosed or something like that. The nucleoprotein has lots of serines and arginines, and so they call this the serine-arginine motif or serine-arginine region, and just a bunch of SR, just like that. But it's not like they're careful, and there's a couple other things in there. The arginines of this motif get phosphorylated in particular. When they're all just arginines, they would have a very high affinity for RNA in general, because they're going to be positive and the RNA is going to be negative. When they are phosphorylated, they are now no longer going to be positive because they have a negative thing stuck on them, and so they would then sort of repel general RNA. But there are other, mostly lysine amino acids that are going to be available, and those are going to do specific RNA interactions. And so the nucleoprotein has a way to bind RNA in general, and then if more specificity is needed down the road, it can get phosphorylated by the cell, and it will then be a little more specific, but less good at general RNA binding. The E protein is going to be palmitoylated at cysteines right after the amphipathic helix, after the transmembrane, and that is a good place to do it. It's very reminiscent of the tail of the spike protein, honestly. And the spike protein is going to be glycosylated at 22 different sites. They are all end-linked glycans at all those sites, and it is going to be palmitoylated, at cysteines in the tail. It's kind of like the envelope protein. Both of these things have to be palmitoylated for the proteins to work efficiently. They do not work very well if there's no palmitate attached. All right, and last one, let's talk a little bit about transport. So transport of the M protein is thought to involve contact with Golgi and Golgi intermediate compartment proteins, and so we think this one is going to be shuttled either to the Golgi intermediate compartment or back and forth between the and the Golgi, so it just ends up in transit in the middle most of the time. It is not clear what motifs are being used for this transport, and it seems to be different on different viruses. Watch this space. The most likely position for those transport motifs would be right down in the tail of M, and that is also the part that's going to interact with the nucleoprotein. And it may be that some of the motifs that are interacting with the nucleoprotein are actually the same ones that are responsible for it being transported. So it's like you can either hold onto a cellular protein and be dragged to the right spot, or you can hold onto a nucleoprotein and then you're no longer going to be dragged to the right spot. The spike the envelope protein is going to interact with proteins, and if the envelope protein is present, it helps to retain spike in the endoplasmic reticulum, even without other factors being there. Spike protein, we at least know more about this. This is a better known motif. It's a dibasic motif, and I think that's the same motif that's going to bind some things like, I believe, calnexin in the but it's also going to interact with the beta subunit of a WD40 protein. And so this is part of the codomer 1 complex. That's going to be the cargo hook, if you like. It's going to bind to the KXHXX motif right in the tail on a little alpha helix, and that is then going to COP1, codomer 1, forms something like a clathrin-coated pit, only it's a codomer 1 coated pit. And it's going to be very important for transport from the back toward the, from the Golgi back toward the So it's retrograde transport. Normally when you're making things, they go to Golgi and out. And that's like the default pathway. That's what will happen unless COP2 is involved, or COP1 is involved, and then COP1 will grab you and bring you back if some proteins have escaped the So the virus is just using this system that was already in place in the cell in order to make sure that its proteins accumulate at the right area. All right, so that's enough about the components themselves. Now we'll talk about how they work in the next section.